Awesome. So thank you everyone for being here. I'm really excited to share with you the work that basically composed the bulk of my PhD and what I want to do in the future. I will start by saying that I know that this is a biomedical audience and the bulk of this talk is going to be about robotics, but I hope I can convince you that robotics can teach us a lot about medicine and convince you that maybe this is something that you might want to work on or integrate into the work that you do in the future. With that, I will get started. Awesome. So I don't need to convince too many people here that biological materials are really exciting and important, but I come at this from the perspective of a mechanical engineer. So as Brian mentioned, I got my undergraduate degree at Cornell. I trained as a mechanical engineer. And one of my, every single one of my classes taught me that engineers build with things like metal and plastics and composites, things that make up everything in our built environment and have done really great for us over the past hundreds of years, and that we plan on continuing to build with in the future. And I was very much one of these sorts of people that was very immersed in traditional mechanical engineering and excited about doing that and applying that towards implantable devices in the future. But my very first job, actually, I'm an immigrant. I was an international student at the time that I was at Cornell. And so a lot of you might know that it can be very difficult to get internships and things like that when you don't have access to those programs. And so my very first job was basically I offered anyone, like, I will do any volunteering experience at all. I just need to put something on my resume. And one of the first jobs that I got was in a biology lab where they were looking at skeletal muscle of rodents. And they knew that if you drink a lot of alcohol, if you're a chronic alcoholic, essentially, your skeletal muscle degenerates. And we also all know from our lived experience that people who spend a ton of time in the gym have great skeletal muscle. They look great and they probably feel great too. So they wanted to see what happens if you drink a lot and you also work out. Can you essentially cancel out the negative and positive effects of those two stimuli? And they needed a poor little undergrad, teenage me, to sit in a basement for several hours a day, every single day of my first summer as a college student, putting drunk rats on treadmills and looking at how the force that they were producing in their legs changed over time. And so, the first month I was pretty depressed. I was like, this is a terrible way to spend my life. What kind of mechanical engineer am I? But as I spent more time in that basement, I think things got pretty philosophical for me. And I started realizing that the actuators that rats were using to generate force and produce motion, which was their skeletal muscle, were so much better than any of the actuators I was building within my classes. Whether you're looking at pneumatic actuators, shape memory alloys, motors, nothing can convert energy into mechanical output with the efficiency and also the adaptability that skeletal muscle and muscle in general can. And so that really got me excited about biological materials. I found out that they can sense and respond to environmental signals in a real time in a way that we can't replicate with even the state of the art in synthetic materials. And this really drove my passion into thinking about whether we could build machines that capture these inherently better qualities of biological materials as functional behaviors. So can they start doing things like self-assemble, self-heal, self-organize purely because they have biological materials integrated with them? So that was the motivation for me to build with biology, or build biohybrid, as it were. And of course, there are many different kinds of machines. You can't build everything. So I, like many other people, tend to love robots. And I think when we have a picture of a robot in our head, a lot of times we think of things that kind of look like this guy, right? Like something you would see in an Avengers movie, vaguely humanoid, primarily made out of, metal and some sort of fancy composite. And definitely, it's a very mechanical system, something that can generate force and produce motion. But really, a robot doesn't have to look like that, as I'm sure you all know. A robot is basically just any device or machine that somewhat autonomously, I can't say that word ever, senses, processes, and responds to its environment in real time. And so a biological robot, or a biobot, is a robot that uses biological materials to perform one or all of those functions. And the hope of integrating a biological material into a robot would be that it would essentially have the same advantages over synthetic materials that we talked about before. It would have dynamic adaptive functionality to a changing environment that synthetic materials and robots just don't have. Now, pretty much every robot needs to generate force and produce motion. And so there were a lot of examples when I started my PhD of robots that used cardiac muscle sheets to do things like swim and grip and all kinds of things. And they were very wonderful. But, they have a couple main disadvantages, which is that cardiac muscle never turns off. Right? So that's great for our hearts, keeps us alive, but not great for a robot because you never want, a TV that you can never shut off. And so you want a robot that you have more control over. The second thing is that they had no regenerative capacity. We know that adult cardiac muscle, when damaged, things like heart attack or other kinds of diseases, can't recuperate itself. And so you don't really have sort of the ability to regenerate, the ability to have a robust system in the same way that you would with a different kind of muscle, for example. So I became kind of an evangelist for skeletal muscle during this time. So skeletal muscle is, of course, what all of us and many mammalian systems use as an actuator. The really nice thing for those of you who don't know, at the microscale, how muscle contracts is you have actin and myosin filaments sliding against each other. Every time they receive a contraction force, this is what's happening at the microscale. But the nice thing is that it's very modular. So even though that's what's happening at the microscale, if you stack many of those in parallel in series, you can generate very large forces. And actually, something that I was very surprised to learn as a mechanical engineer is that muscle, if you combine its size and weight as well as the size and weight of its power source, which is essentially just sugar water with some amino acids in it, produces larger strains from smaller volumes than most synthetic actuators. So this is a really desirable power to weight ratio. It's biodegradable, and also it can heal from small volume damages. And the best part is that there were already some very nice established protocols for building skeletal muscle or tissue engineering skeletal muscle in the lab. So it's a great starting point for us and something that I decided to use in my future work. So building a skeletal muscle powered robot is, I don't want to say a simple process, but maybe simple to explain. So in our bodies, muscle is tethered to our bones via stiff tendons and stretched across an articulating joint like the elbow. Every time it receives a signal from a motor neuron to contract, the muscle does so, and we're able to generate these large locomotions. And so we can essentially recreate that in the lab by 3D printing a low stiffness beam that mimics that articulating joint, at the two ends of which we have these high stiffness pillars that mimic the tendon. And around that, we want to anchor what we call our muscle. So to make the muscle, we pursued kind of like a modular approach. We injection molded a solution of CTC12 skeletal muscle myoblasts mixed in with fibrin, thrombin, matrigel, normal things that sort of mimic the extracellular matrix inside the body. And again, biology does a lot of the work for us at this point. So at this point, all I'm doing is kind of just making jello. And then I let the cells go. They proliferate. They exert traction forces on their surrounding matrix to compact into a dense 3D strip of tissue. The individual myoblasts fuse to form myotubes, multinucleated fibers that are the basic contractile unit of muscle. And you essentially get something that ends up looking like this in a schematic, or here they are in picture. So these are about 6 millimeters long. I'll talk more about scale later. But essentially, the nice thing about this is that you can pick up these injection molded muscle rings and transfer them to skeletons of a wide variety of shapes and sizes. So here I'm just showing it transferred to a 2-legged skeleton that we're going to use for walking. The important thing to note here is that already you start seeing that there's a huge difference with building with biology. So if you change any aspect of this cell gel solution and composition, you can have muscles of vastly different sizes and strengths and compositions. The other thing that we see is that the mechanical environment also offers a ton of cues for the muscles. So if you culture these muscle rings or rubber bands on stiffer skeletons, they are able to produce more force. If you culture them on softer skeletons, they produce less force. So this is something that you would never see with a synthetic actuator, where the environment is highly guiding the force and the form of the robot over time. So the force is greater for a couple different reasons. One is that the mechanical cues themselves signal to the muscle that hypertrophy needs to happen. They're being placed under higher loads, and so they start maturing at a faster rate and massively upregulate some mid and late stage myogenic markers. And so they're bulking up for that reason. But the other reason is that the force being imposed on the muscle by the skeleton provides a cue to also align the fibers in the same direction. So if you had a muscle tissue clump just sort of sitting there with no sort of mechanical environment, you would get probably fiber formation, but those fibers would not be aligned with each other. So it'd sort of just be a ball. But if you stretch it around a skeleton in this way, you can see that in the middle, where it's getting this this sort of stretch stimulus from the skeleton, all of the fibers are highly aligned and parallel to each other. And so all of their individual contractile forces are summing up in the same direction and producing an observable contractile force, as we'll see. The other important thing to note, if you look at a cross-section of the muscle, the green fluorescence here is a myosin protein, so that's a marker of mature differentiated contractile muscle. It's expressed uniformly throughout the thickness of the tissue, and again, follows the tension cues that you're getting from the robotic skeleton. So this is what it looks like on this particular robotic skeleton. If you put it on something different, it would align differently. All right, so before this video plays, I want to give it away all of a sudden. So in our bodies, of course, the muscle receives contractile stimuli from motor neurons, which is essentially a chemical, but really an electrical stimulus, right? It's opening up calcium ion channels. The ions are flowing in. The muscle knows that it is getting an action potential that's telling it to contract, and it contracts. We obviously don't have that in a simple engineered muscle platform in the lab, but we can replicate it in a couple different ways. So our very first paper on this work was published in 2014, actually, now in PNAS, where all we did was we took these biological robots and placed them between two electrodes, sent an electrical pulse through them, saw the muscle contract, and we could get it to do a few different things. Since we have a short time today, I'm not going to show that. What I will show you is our next generation robot that we published in 2016, also in PNAS, where we were able to control the force production of these robots using optogenetics. So we took our C2C12 myoblasts, genetically engineered them to express a mutant variant of channel rhodopsin, basically a calcium ion channel that opens in response to 470 nanometer blue light. And we showed that if we essentially just flash blue light pulses on these robots, we can get the muscle to contract. So you see the skeleton. That central whiter portion is the muscle tissue. And every time the muscle flashes on it, you can see the muscle contract and the resultant skeleton deform. So this is very repeatable across multiple robots. And what you see is that if you do 1 Hertz stimulation contraction, you get contraction, relaxation back to its passive state, and then contraction again, relaxation back to its passive state. As you increase the frequency of stimulation, the muscle isn't able to fully relax. So you'll get contraction, relaxation, contraction again, relaxation, and it does this up until about 10 Hertz, at which point you get muscle tetanus. So that happens physiologically in our bodies and also happens in these engineered muscle constructs. If you overstimulate it, they essentially get to a place where they're contracting so much that they can't really move anymore. So this is all very encouraging. It shows us that robots are acting the way they should be. And also were the first demonstrations, both that an electrically stimulated pulse as well as a light stimulated pulse could be used to make an untethered robot that was actuated by engineered skeletal muscle. Now, the interesting thing about these is that you can also get them to do different types of functional behaviors. Today I'll talk about locomotion, but other folks have used the same sort of modular ring-based actuator to make things like grippers or pumps, etc. Essentially, it's just something that converts chemical energy into linear actuation, so you can use it for a wide variety of different functional behaviors. But to make something that walks, there's a few different ways you can do this. One particular type of walking robot that we made is a simple 2-legged structure that's completely symmetric and powered by 2 muscles. And we assumed that because we had this sort of spatiotemporal control of where the skeletal muscle contracted because of this light stimulus, if you stimulate only 1 leg, ideally the robot is going to move in the direction of the stimulated leg. And if you stimulate only half of a leg, you see rotation of the robot in the direction that you specify. So lots of flashing lights coming your way. So on the left-hand side is a robot that's where you have stimulated 1 whole leg, and you see slow locomotion of that robot towards the direction of the stimulated leg. And when you stimulate half of the muscle ring, you get rotation. And so with these 2 motions of directional locomotion and rotational locomotion, you can essentially map out an entire 2D space and trajectory with your robot. It will walk in any direction that the light tells it to walk. So this is all well and good, and hopefully I've convinced you that if nothing else, they're cute. But nothing about what I've shown you so far is, regardless of being the first demonstration or whatnot, is doing something that a synthetic robot can't do. right? So any synthetic robot can generate force and produce motion and walk around, and can probably do it faster than my robots can. But what my robots can do, or biobots can do that a robot can't do, is adaptively respond to their environment, both to loss of function as well as gain of function cues. So going back to one of the adaptive functional behaviors we discussed before of, we can heal and robots and out in the wild can't, we can do that for a variety of different reasons. So specifically for skeletal muscle in the body, the way in vivo damage is dealt with is that the actual myofibers themselves are terminally mitotic. So they can't differentiate, and they've differentiated, which means that if they are damaged, they are irrevocably damaged and they can't repair themselves. So what they do is we have these muscle stem cells that sit outside of the muscle fiber, proliferate, they locomote to the site of damage, proliferate, and then fuse to form new fibers or fuse with existing fibers to restore force production in the damaged muscle. So we wanted to see if we could replicate something about that process in our biological robots. So first, of course, we need to create a model of damage. So the way we do this is make the robots just as before, but then go in with a pair of micro scissors and do the unthinkable. Culture them for 2 weeks and then damage them. And so what you get is something like this lacerative tear. The reason we chose this sort of tear is it's very similar to the predominant cause of damage in our body is to muscle, which is mechanical tears due to overstrain or injury. So what you see with these tears is that it matters very much where you tear it and how much you tear it. So as you can imagine, if you tear it in this middle region where it's experiencing a lot of tensile forces, you'll see that that tear will propagate over time and essentially the tissue will rupture. Over a period of several days, and the time to rupture depends very much on how much you damage it. So as you can imagine, if you cut it more, it ruptures more quickly, and it also produces less force. So you can see that there's a pretty strong correlation between how much of the original cross-section of the muscle you have left and how much of the force the muscle is producing as compared to healthy tissue. However, even though there is a significant drop in viability between healthy and damaged tissue, the relative viability is not that bad. And that also makes sense. You're damaging a lot of fibers in the most crucial force-producing area of the tissue, but you're not actually damaging a ton of cells as compared to the whole volume of the tissue. So there's a significant drop in reduction in viability, rather, but it's not huge. However, this drop in performance is not something that can be recovered simply by healing the tear. So if you go in, you tear the muscle, and then you essentially just heal it with a biological sort of fibrin-based glue, you would imagine that if there were enough viable cells in this area, as long as the tear wasn't allowed to propagate, the tissue would eventually be able to regenerate force at its previous levels, or at least something like its previous levels. But this turns out not to be the case. So if this is the amount of active tension or contractile force, I don't know why there aren't units on that, but it should be in micronewtons, that healthy tissue is producing. This is what happens when it's damaged at about 50% of its original strength. And when you heal it with a glue, you can see that while it might not rupture anymore, it isn't really recovering its force production. And what you see is actually that to recover force production, what you need to do is replicate what exists inside the body, right? So in our body, we're recruiting stem cells. So in the same way, if we mix in some undifferentiated myoblasts with our biological glue and add it to the tear, we see slow recovery of the force production as the cells start fusing to form contractile fibers, and also upregulation of some early and mid-stage myogenic markers. So that's well and good, but it's still obviously less than what healthy tissue is producing. So we go back to the idea of different types of adaptation. So yes, we are able to adapt to loss of function cues like damage, but we also need to be able to adapt to gain of function cues like exercise. So revisiting the idea, say, of the rodents on the treadmills back in the day, I figured, there have been plenty of examples of people using mechanical stretch stimuli or electrical stimuli to exercise skeletal muscle. And we can do the same thing here by using light stimulation instead. So we showed actually for the first time that daily stimulation of the skeletal muscle during the period of differentiation using an optogenetic light stimulus slowly increased the force production that the muscle was able to produce over time. And that if we combined only optical stimulation with optical plus mechanical stretch stimulation during differentiation, you would see a synergistic increase in force production as well. And the other interesting thing that we noted is we wanted to know whether it was important to stimulate the whole muscle, just the newly added muscle, or the existing undamaged muscle. And we found that actually, because the cells were not only fusing to form new fibers, but also fusing with existing fibers, it was really important to stimulate both the newly added tissue as well as the existing tissue with light in order to completely recover force production after damage after about 7 days. And if we wanted to accelerate this healing process even further, in our cell and glue mixture, we also mixed in some microparticles containing human insulin-like growth factor, or IGF-1, which we'd shown in the past could accelerate maturation and hypertrophy of our skeletal muscle. And we saw with that, we got recovery of force production that was not significantly different from healthy tissue within about 2 days post-damage. When we look at the protein expression levels in those tissues, we see massive upregulation of mid and late stage myogenic markers. So the contractile apparatus is completely reforming. And we actually take sort of a fluorescent cross-sectional peek into the tissue. This red tissue is the original damaged muscle. The green cells are what we've added in the biological glue that proliferate over the site and repair the tear. And then you see expression of myosin throughout that whole tissue section. So I just want to wrap up by saying some of the things that we're working on right now and hope to work on in the future are incorporating other types of cells. So endothelial cells for being able to scale up these robots, neurons for being able to have process more stimuli than just on off, but make some sort of minor decisions or respond to things like chemicals and pressure and temperature. We're also looking, we've done a lot of studies on controlling the lifetime of these muscles. So not only their cryopreservation and revival, which we can do very well, but also essentially embedding something like a kill switch in them, where we would be able to turn them off at will and tell them to biodegrade when they're no longer required. I've done a lot of work of integrating biobots and biofabrication curriculum into the curriculum both at University of Illinois and MIT. We teach students to design and build their own biological robots. And some of the work that I'm working on with Bob right now is how we can start thinking about integrating these sites of biological or biohybrid machines and robots as implantable devices that respond to individual needs. Because as you can imagine, the place where we need this sort of dynamic control the most is in the very dynamic environment of the human body and in applications that require precision medicine. So I know that this was a robotics talk, but some of the things that I've shown you, I hope convince you that in addition to taking inspiration from biology to build our machines, we can also use the machines as mesoscale models of understanding how the body works. So a lot of the healing study that I showed you, yes, it's the first example of a robot that can heal from damage, but we're actually taking that back now to looking at volumetric muscle loss in animals and thinking about how we can do targeted optically based exercise in order to restore muscle force production and control in actual animal models and develop new healing strategies that enable us to do so. So it's kind of a crosstalk. We can use the robots as robots, but we can also use them as mesoscale models of orthopedic systems in the lab. So with that, I'd just like to wrap up by saying that this is a huge cross-disciplinary effort, so much so, and involves so many labs that I didn't have space to put all of the students and postdocs that contributed towards the papers that I showed here, but here are the labs and the institutions they're from. Very grateful for my funding sources, and I wanted to say, if you're not really in the space of tissue engineering or muscle and just want a broad overview, I wrote a short essay in Science last year that covers everything that I talked about in this talk and more. If you are a hardcore tissue engineering muscle person and want to replicate this in your lab, this work was recently featured, or a couple years ago featured on the cover of Nature Protocols, where we tell you everything that you would need to buy to replicate this on your own, including which pair of tweezers works best for me. And I have very strong feelings about that. So yeah, that's all I got.
That was wonderful, Ritu. Thank you so much for that really engaging talk. There are some questions rolling in, so I'd be more than happy to read them to you and you can answer them. So we have a question from Akila. Could you tell, so 2 questions. I'll start off with the first question. Could you tell me a bit more about the maximal skeletal loading possible for a given cultured muscle tissue? And how does this relate to the scaling of these constructs?
Yeah, I think based on my understanding of the question, if you're asking about the forces that the tissue engineered muscle can produce in itself, we have the tissues that we created that we were able to exercise actually produce some of the highest forces that tissue engineered skeletal muscle has demonstrated to date. However, that is significantly less, like 5% or so of what natural muscle can produce. A big part of this is actually a problem with our cell source, in my opinion. So we use C2C12 skeletal muscle
Right.
cells because I was a mechanical engineer. They work really well for people who've never done cell culture before. But now, since I've started my PhD and the time that I've started my postdoc, people have developed some really nice protocols for differentiating skeletal muscle from human-induced pluripotent stem cells that are working really well and that are contractile. So in my future work, I'm transitioning to those and hope that we'll be able to, in combination with that cell type and our culturing and exercise protocol, be able to recuperate something that's much closer to what native muscle can produce.
That's great. And the second question is, you mentioned mechanical and optogenetic stimulations. Have you looked into any electrical stimulations during adaptive exercise regimes?
Yeah. I mentioned mechanical and optical. We've also compared it to electrical and then compared sort of synergistic passages of both. Based on what I can tell, even though we're using different ion channels for electrical versus optical stimulation, you see a pretty similar response to both electrical and optical stimulation. I don't think you see much synergy when you combine those two things. The synergy comes when you combine mechanical stimulation because the way that it's affecting force production is primarily through things like regulating the upregulation of different proteins, but also causing more alignment of the fibers. So electrical and optical stimulation work fairly similarly. Mechanical stimulation works a little bit differently. And some people, I haven't done this, but some people have even looked at active perfusion through the muscle, and flow can also impact force production a little bit as well.
Sure, sure. That's great. We actually, James messaged me privately and mentioned that he had a question, so he can go ahead and ask his.
Thank you. Yeah, I'm very interested in this topic from a personal point of view. But my question was actually more about the blue light scattering.
Yeah.
So because you're using a low wavelength of light, you're going to get quite a degree of scattering from the hydrogel and from the cells, particularly as it matures. So I wonder if you see sort of differences in actuation as you go through the depth of the tissue, as you sort of follow it along the direction of the irradiation. And I wondered if this would be sort of a negative thing, but I also wondered if you could use this as like a non-linear actuation. I saw your construct seem to be sort of lifting up in the middle as they were actuating. I wonder if that was why, maybe?
Yeah, no, this is an excellent point, and something that we think about a lot. So blue light, as you mentioned, does die off very deeply as it penetrates into tissue. So blue light at the wavelength that we're using only penetrates about 550 microns till it dies off to something insignificant. So we have seen things where if we build muscle tissues that are, say, a millimeter, and we pit it against electrical stimulation, the electrical stimulated bots perform much better in that they're generating more force and producing more motion because you're exciting all of the contractile fibers within. Once you get to that 500-ish micron threshold, they perform exactly the same. So there is a thickness dependence. So we can deal with this in two ways. I think one is maybe utilizing it, as you said, you could utilize it as a strength because it offers you greater spatiotemporal control over the subvolume of tissue that you're actuating. So when you're thinking about things like robots being deployed in different settings, you could have implanted optical fibers and stimulate only certain regions of tissue. That can work out very nicely. However, if you're looking at something like repairing volumetric muscle loss and exciting that entire volume of tissue, we would want to be able to do something that was stimulated by far red light or something like that. So there, I think it's kind of more of like a biological issue. I've had a ton of folks try to incorporate things like archaeorhodopsin and halorhodopsin instead of channelrhodopsin into my cells. They've never been able to get it to contract, but if someone can do it for me, you would be my favoriteest person ever. So something to think about.
Awesome, Ritu.
Oh, thank you. That's great.
Maybe if you could answer for me very quickly, have you thought about 3D movement? So you mentioned two-dimensional movement with the kind of biobots that you've been able to build in the lab. And I know of those, those tiny little octopuses that like
Mhm.
people have made. So I don't know if you have been able to make 3D movement with your biobot systems, or have you looked at
Yeah,
it?
so I haven't, but my co-advisor's lab and people out of it have used that sort of ring technology at different scales and in different designs. So we have some people that do sort of pumping in 3D, but also we have people who have made these sort of sperm bots powered by the skeletal muscle rings that can accomplish 3D swimming as well.
Awesome.
They primarily do it in 2D just because it's easier to observe and track the force production, but it could potentially be used in that domain. So yeah, so that's been really nice for me as, you know, part of this protocols paper, it was like nice to have my name on a nature protocols paper, but the really best part of it is that people were able to take it, reproduce it at different institutions and apply it to different applications, and served as sort of like a gut check for me that it can work in different settings.
Sure, that's awesome, Ritu. Well, thanks again for the wonderful talk and for answering all of our questions.